All right, hey guys, this is our third module for the Intro to Neurotech course, and this third module is going to be about EEG systems and event-related potentials, also known as ERPs. Um, the headsets that we're going to be working with as part of the club, and the headsets that are going to be given to us by Muse and other companies are primarily EEG headsets. So it's a good idea to understand the science behind them and what the data that we're getting actually means, so that we can use this to understand what we can and cannot conclude from it. So going off of what Mateo's module was last week, and just a little recap, the resting membrane potential inside the cell, inside a neuron, is negative 70 millivolts. And if you guys remember from last week, we talked about action potentials for a little bit. And those are the discrete voltage spikes that travel from the cell body to the cell axon, or to the axon terminal. And when I say discrete, I mean that it's either on or off. There's no in between. The axon either fires or it doesn't fire. And these axons are millisecond duration, so they happen almost instantaneously, and they set off kind of a chain reaction down the whole body of the axon terminal. The problem with this is that because they fire so quickly and because they last only milliseconds, they also don't fire at the same time. So they don't, to the human eye, I guess you could say that they fire at exactly the same time, but they're not within microseconds of each other. And so the problem with this is that they often fire in different orientations, because, like, the neurons and everything in the brain are in different networks, and they're all scattered all over the place. So you'll have axons firing at different angles and different orientations. And because they don't fire within microseconds of each other, they often cancel each other out. And so we can't detect them from just the outside of our head or from the surface of our scalp. But instead, if you look at postsynaptic potentials, and these postsynaptic potentials are basically the result of the neurotransmitters binding to the receptors of the postsynaptic cell. So if you remember, you have the axon, you have the whole cell body, and you have the axon terminal, and you have synaptic potentials going from the axon hillock to the axon terminal. And then once that chain has been initiated and completed, between two neurons, you have a presynaptic neuron and a postsynaptic neuron, and in the middle, you have that synapse. And so when a neurotransmitter gets released in that intermediate space and then binds to the other neuron, that's called a postsynaptic potential. And those, compared to action potentials, last for tens or hundreds of milliseconds. And that's a good thing for us, because they often do fire in similar, within microseconds of each other. And they're often oriented in a way that is almost homogeneous. So there's a certain group of cells called pyramidal cells in the brain, and those are all perpendicular to the scalp surface. So they're all kind of like right angles, jutting, I guess, inward if you were looking down. And because they're oriented in such a uniform way, when they fire, their signals can be summed up. And the summation effect is what we can detect from the outside of the scalp. So when you put an electrode on someone's head, what we are detecting is the postsynaptic potential, and not the postsynaptic potential of just one neuron, but a population of neurons. So we'd be able to detect when like hundreds or a couple thousand neurons fire at the same exact time. And this is good, because what we want to see is the effect of something. So when we do a particular task, when we pay attention to something, or when we see something in our visual field, oftentimes that's strong enough to elicit a response from a group of neurons or a network of neurons, and that is exactly what we want to analyze and understand the effect of. So now that we've kind of covered that, a little bit of recap, because we're studying primarily electrical signals of the brain, it's really important that we have a strong understanding of the basic principles of electricity and how they relate to each other. So the first one that's really important is current. And current is the actual flow of electricity through a conductor. People often confuse current and voltage, and I mean, before I was even involved in this stuff, I definitely did not know the difference. But current is just the actual flow of electricity through a conductor. So it's the number of charged particles, whether that be electrons or protons, that pass a given point in a specific amount of time. So it's like measuring the rate of flow, basically. And speaking about the rate of flow, you can basically think of it as like water going through a pipe. So just like when you turn on a hose and water flows through the pipe, and you can measure how much water, the rate of which water is flowing, or how much water is passing through a given point in the pipe at a given point in time, you can also measure current with those same kind of metrics. Voltage, on the other hand, is also called electrical potential, and voltage is the potential for current or electrical current to flow from one place to another. So down here, I have a picture of the UC Davis water tower, or like one of them. Um, and that's to symbolize like potential energy for water flowing. So just how you know, like if you have water flowing from an elevated surface to a lower point anywhere, there's always going to be potential energy, even if the water is not flowing. That's what voltage is. It is the measurement of the potential for the electric current to flow from one place to another. And notice that because it's measuring potential energy, it's always relative. So it doesn't make sense when you say like the voltage at a specific point or a voltage of a particular electrode. It's always relative, like relative to what? What is its potential between two different things? So just keep that in mind as we continue. So what exactly is an EEG? So EEG stands for electroencephalogram or electroencephalography. It is the measurement of electrical impulses in the brain. So the brain, or I mean, all muscles in our body to a certain extent are electrical organs. They give off, they're conductive. They give off electrical signals. So the brain obviously is the hub for all of those signals, and it's the measurement of all these electrical activity going on in our brain. So I'm actually a research assistant in a cognitive neuroscience lab, and this is the technique that we use to record our data. And so a little bit about how it works is that you basically, I mean, if you're a subject, if you go into the lab, what they do is they measure the circumference of your head to determine a particular cap size that would fit you best. And then you sit down and they put that cap on your head. It's kind of like a mix between a swim cap and a shower cap. And on that, we place electrodes. And there are different numbers of electrodes you could place. Some studies use 32, some use 64, and some use 128. So depending on what you're trying to find out or what you're trying to research, you place the amount of electrodes that you want. And that takes a decent amount of time, like 64 electrodes on a person's head. You have to literally go and place each one in the right location. And then after you've done that, there's a gel-like substance that is typically used. And the gel-like substance is basically a mixture of salt, which is like we need something that can basically conduct the electricity between the scalp and the electrode. So obviously your scalp is not completely flat, and you obviously have hair in the way. So you want to make sure the signal is as clean as possible and the electrode can grasp as much of the signal as it can. So you usually use a gel-like substance, the primary ingredient, which is salt, between the electrode and your scalp. So that helps to hold it in place. So after you place all of those electrodes, you go in with a syringe and you basically squeeze gel in between each 64 of those electrodes. And you let it sit for a bit so that it can solidify, hold the electrode in place, and you have a good conductive signal. So if you look at this guy right here, this is kind of what it looks like. It's super crazy. Like you just have a bunch of wires all over the place and a bunch of things on your head. On the right here is what we call the 1020 system. So you don't just place the electrodes randomly. There's a certain code. There's a certain system for that in place. And so this right here is what's called the 1020 system, and it's also internationally followed across the world. And if you pay attention closely, these things at first glance might seem kind of gibberish, like they don't make any sense, but they're actually not terribly hard to follow, and it's kind of intuitive if you know a little bit about the brain and the anatomy. So if you look at C over here, C is the central, right? C refers to central. So this is, if you imagine this as like a 3D surface, this is kind of like at the very middle, and then you have it going laterally across the side. And then you have P right here. P refers to the parietal lobe, right? F right here refers to the frontal lobe, and you have the T's on the sides, which refers to the temporal lobe. So if you remember from module 1, they're the 4 main lobes of the brain, and so these electrodes are organized to fit across main areas of the brain. And obviously, if you see a combination of letters, that's like central parietal. So it's between the central and the parietal lobes of the brain. So it's fairly intuitive if you know a little bit about the brain structure. So one other thing you should know about EEG systems is that there are 3 kinds of electrodes. There's an active electrode, there's a ground electrode, and there's a reference electrode. And each system, every single EEG system has all 3 electrodes. We'll go into detail about what the difference between these electrodes are, but just keep in mind they're different kinds of electrodes, and they serve a different purpose. Also, another thing to make note of is that the brain is an electrical organ. So whether or not you're doing something intentionally, or you're just sitting there, or if you're just even sleeping, there are electrical signals and there are electrical signals being released all the time. And so this technique is most advantageous when you use it paired with a certain controlled stimulus in a controlled experiment. And here's what I mean by that. So if we put an EEG headset on someone's brain and just made them like sit there without doing anything, you would still see a bunch of activity. In fact, if you look closely at this picture, this is basically what you see when you're looking at an EEG headset. Every single electrode is picking up activity, and you'll just see lines of like 64 lines or the 128 lines of just microvolts. And it's hard to tell what that even means when you're looking at it in its raw form. But however, let's say we have this baseline recording of them just sitting there, and then you suddenly show them something and you see a spike. Well, that's an event-related potential. And the name makes sense because it's an electrical potential that is related to an event that has just occurred. So these event-related potentials are a result of direct sensory, cognitive, or motor movements, and that is exactly what we study when we use an EEG system and look at electroencephalography. We compare the baseline activity of just someone sitting there to the ERP, the event-related potential, to see what is going on inside the brain when a particular stimulus is presented or the task is performed. So it's all relative. If you see, we kind of subtract what we see, what the stimulus generated from the baseline, to get an idea of what is going on. So what I showed you just in the previous slide, I'll show it once again, is the traditional EEG system. This is what is used in like clinical laboratories or research laboratories at universities and hospitals. What we're transitioning to in the neurotech industry are dry electrode BCI EEG systems. So this is actually a product of a company called Zito. And Zito is the first company in the US that's trying to make a dry electrode system that is FDA clinically approved, and they've actually gotten their approval. So I actually had the opportunity to talk to the CEO of this company a couple months ago while they were making this product. And I was just trying to understand, how this is different from a traditional EEG system, because as you can see, first of all, it looks super different. But I wanted to kind of understand the technical differences. And so for the first most important difference is that they're dry electrodes instead of wet. So they don't use any sort of gel, they don't use any sort of substance, it's just dry electrodes. And the electrodes can come in primarily 2 different shapes. The first one is kind of like the teeth of a comb. They're like kind of prongs that are not sharp, but they find the way through your hair, and they kind of just rest on your scalp. The other are like foam pads that you can put, and those are kind of for areas of the head where there is no hair, like your forehead or your like temporals right here. And so they're dry electrodes. Secondly, in this particular device, there are only 19 electrode channels instead of the regular 32 or 64 that you might see in a clinical setting. Those are the primary differences. But But what this kind of leads to is that are you compromising accuracy and signal quality by having less electrodes? To a certain extent, it's more convenient, especially if you want to use this because A, it takes a long time to put gel and stuff on a patient in a clinical setting. And even in the lab, like for me, it takes an hour just to set up the stuff. Having a process like this is super easy where you just make them wear the device, and so it can speed up a lot of things, but you might also be um, sacrificing accuracy and signal quality. And while it has been FDA approved, there's a lot it's definitely not as accurate as a clinical setup, and it definitely cannot pick up the subtleties that a clinical EEG system could do. And so um, in the neurotech club, or like a lot of in the neurotech industry, a lot of what we're going to be working with is dry electrode stuff, and it won't have that many channels. Like the Muse headset, for example, only has 6 to 8 channels. So there's limits to like what you can make with it and how much data you can gather. And there's also limits to like how accurate the data is that you're gathering, but it's at the trade-off is expense and convenience. So these devices are cheaper and they're faster and they're easier to maintain. So that's the trade-off that we kind of face right now between industry and academic settings. So now going on to um, active, reference, and ground electrodes. So every EEG system has an active, reference, and ground electrode, and every circuit has a ground electrode. Um, in physics, I won't get into this too much, but you use grounds to like you might have heard of this, but like in our houses and any sort of like big infrastructure type areas, there's always a ground in case um, the circuits don't function properly, or like something gets tripped. Instead of like electrocuting or spontaneously combusting, they run it into the ground, and it's much safer that way. And so um, in an EEG system, when a voltage is measured between two electrodes, one of them must be connected to the ground circuit. But if we connect the ground to the earth, like in normal circuits, the voltage would reflect any surplus charge that's built up in the subject, and that static electricity would basically overpower the neural signals, because the signals from our brain are so subtle, it would basically be lost in all of that. There's another alternative where we can have an electrode on the subject, which is grounded to the earth, or which is connected to another circuit that's grounded to the earth. But that is dangerous if it isn't grounded properly for obvious reasons. Um, so what usually happens is that an EEG system creates a virtual ground. It, it creates a virtual ground, and we have a ground electrode that is connected to that ground circuit. And that problem is kind of solved there. There's a lot more nuances and like honestly a lot more technicalities, but for all intents and purposes, that's pretty much all you kind of need to understand about that at this point. So here's how those three things are related. So like you have your ground electrode, which is a necessary component of just circuits in general. But as I said before, the way the EEG system is used, or the ERP technique is used, is that it's all relative. So you have some sort of baseline, and you have some sort of stimulus that you show to someone, or some sort of event that occurs that a subject witnesses or takes part in, and you subtract that activity from the baseline activity to look at what's going on and what the differences are. So with that being said, we need like some sort of reference. Ideally, we would like a reference point that doesn't have electrical activity, so we can look at what's happening in complete isolation. But they found this out much later on, but there's no electrically neutral point in the human body. So that's what I mean when I say no Switzerland principle. This is actually a term that my PI taught me in the lab. But what he means by this is that there is no neutral point. Switzerland's been known to be politically neutral, and taking an analogy from that, there's no electrically neutral point in the body. Everything has electrical activity that can be picked up by the electrode in some form or fashion. So what the idea between an active, reference, and ground electrode is that voltage needs to be measured between two electrodes. As I said earlier, voltage is like is always relative. It's the potential for a current to flow between two electrodes. So in this case, the only way to measure the potential energy is involving the ground electrode. So if you want to measure the voltage at an active electrode, you have to do active minus ground. And if you want to measure the potential or the voltage at a reference electrode, you have to measure the reference minus ground. But you also want to end up with subtracting the active from the reference in order to actually isolate the component that you're looking at. So what we do is we do active minus ground and then subtract that from reference minus ground. And if you do the math, you'll see that the G's cancel out. And so what you do get is just A minus R. So this is just simple algebra, but it proves the point of like us being able to isolate what we're looking for while dealing with the complexities of a ground circuit and having that in an EEG system. And so this is typically how everything is done. And the idea for a reference point is that we want to pick a point. It doesn't really matter where in this case, because no point is completely electrically neutral. But there are certain tips for choosing a good reference point. So conventionally in an ERP system, we usually use the mastoids, and the mastoids are like the bony parts right behind your ear right here. We also use the two temporal parts right here, and these are helpful in detecting forehead movements, like eyebrow movements and eye blinks, which are often associated with a lot of stimulus presentation. So you always blink, and moving your eyes can tense up your forehead muscles, which will also release signals. So having a reference from that area is always a good idea. And so when choosing a reference, you want to choose something that's convenient. You want to choose an area that's not biasing one part of the head from another. So if you're choosing to use one right mastoid, you should probably balance it out on the other ear as well. You don't want to put it where there are too many big muscles. So you don't want to put it near your neck or near your jaw, because those are huge muscles that actually cause a lot of distortion in the data. And you want to avoid placing a reference where the component is obviously going to be strongest. So if like there's a strong thing that you're looking for near the occipital lobe, you probably shouldn't place a reference in the back of your head, because the idea is to try to isolate the component, not make the reference as similar to the active electrode. One of the things that is really useful in neurotech devices is that the data can be referenced online and offline. So the Muse headband, as well as other neurotechnological devices, come with references in them and reference electrodes. And I'm not too sure whether you can change the location of the reference electrodes or whether that's kind of done for you, but there's software to automatically read reference it. So that's always a plus, and that also allows a lot of flexibility. So why do we use an EEG system in the first place? There are other methods to use that are being used in clinical laboratories, like MRIs and PET scans. And so this table gives a good overview between all the different types of measures and the pros and cons. So starting off in the first column, you have the microelectrode measures. So those are like literally using cranial electrodes to go inside of your brain and actually detect singular neuron firing rates. And so it's very invasive, which is why it's kind of poor in this category. Its resolutions are excellent because it's literally in the brain. So you can get a great idea of like where exactly you are and like how fast and stuff is happening. It's obviously very expensive as well, because like it literally requires surgery for that to happen. There are other measures like the PET scan and fMRI, and these are very good for spatial resolution. So you have a good idea of like where the blood is flowing in the brain. That's basically what this tells you. And so you get these nice, like pretty pictures of blood flow. But the problem is blood takes time to flow in those areas. So you have bad temporal resolution. It's a bad time scale. This is where EEGs actually excel at. So they're not invasive at all. It's just something being put on the surface of your head. And their spatial resolution is not too great, because that's really not the point of an EEG. You're not trying to figure out where the signal is coming from. You're just trying to figure out the difference between things happening. So the resolution, the temporal resolution, is actually fantastic. It's down to the milliseconds. And so we are really able to look at how our brain changes immediately at the onset of a particular stimulus. So one big I guess principle of electricity and electric signals in the brain is volume conduction. So when a dipole is present in the brain, the current is conducted through the medium until it reaches that surface. So um, this is why it's called volume conduction, because current is conducted through the entirety of the volume. This can be analogous to like electricity in a power plant. So when electricity is generated in a power plant, and then that electricity goes through power lines to people's houses and businesses, those same electrons in the power plant don't go from the power plant to your house. It's not the very same electron. Instead, one electron pushes the next, and so on and so forth. In the same way, when a dipole is created in the brain, there's no like lag time to wait for the charged particles to move to the surface. There's like an instantaneous voltage that is created and then propagated from the site of where it's generated, or like we call that the generator, to the scalp where the electrode is actually picking up those signals. So this means that the ERPs are distributed throughout the scalp instead of just one electrode. So when you're actually having like 64 electrodes on your head and you're having all these components, ERP components being generated, it's not like one electrode picks up one kind of component. It's more like something is generated in your head and it disperses and spreads out through the entire medium, which is your brain, through the principle of volume conduction. And these electrodes, which are on different parts of your head, pick up the same signals, but they pick it up to varying degrees of intensity. So for example, if a signal is generated at the front of my head in the frontal lobes, obviously the electrodes at the frontal part of my brain or the frontal part of my scalp will pick up those signals at a higher degree of intensity than the electrodes on the back of my head. It's fairly an intuitive concept, but it has large implications and it's easy to forget when you're actually analyzing data. So keep that in mind. One of the problems with this, though, is that it leads to the superposition problem. So to explain a little bit about what is going on in this diagram, if you look right here at this guy's head, you have different ERP components being generated. So you have C1, C2, and C3. And they're all in different aspects of the brain. So these are called generators, and these are called generation sites, right? And then over here in E1, E2, and E3, you have electrodes. This E3 looks like it's in the center of someone's head, but it's actually in the back of someone's head. Just keep that in mind. So you have all these electrodes and you have three different components. And what this is saying is that the voltage, the waveform that you observe on E1 is not a direct wave generated by any one of these. Instead, it's a combination of it. So you have a little bit of C1, a little bit of C2, and a little bit of C3, and there's a weight factor. So maybe like 10% of this and 30% of that and 40% of this is what you see as a wave at the first electrode site. And the same thing, except a different linear combination, is what you see at the second electrode site, and so on and so forth. So if you're looking at this, you might think that this is kind of like a neural network model where you have the connection and the weights. And yeah, that's actually pretty accurate in the sense like that's a good way to think about it. It's not as easy to decode, and you can't exactly train it like that in the same way like you could train a neural network. But the idea of connections and weights holds. And this is a very key idea to keep in mind, because people often think, at least when I first started, I thought that like whenever you have an electrode, what you're seeing is supposed to tell you something, and each electrode means something different, but it's all just like a different combination of like what's happening in your head. So all 64 electrodes that's on your head are representing different components at different intensities. And the biggest problem in EEG research and EEG studies is that there's no foolproof way to recover the underlying components from the observed waveforms. Like you can't isolate it because these weights and these connections are so random and they're not consistent. Forget being consistent from person to person, they're not consistent from trial to trial in the same person. So it makes it really hard to understand, like, like to what extent am I seeing this component versus this component, and what's the influence of the stimulus on each one of these components. Um, so to put in words what I just said, each electrode picks X% of a source waveform in the brain, and that percentage depends on the position and orientation of the generator relative to the electrode. So like I said, if the signal is in the front of my brain, it's obviously going to be picked up with better intensity and be represented at a higher percentage of, of that electrode waveform at the front of my head than it would be if that electrode were to be in the back of my head. So the observed waveforms are a combination of several underlying components. And just to reiterate again, it's like extremely hard to isolate components from one another. And that's the big impediment that we have for EEG research right now. So another issue or another kind of situation about EEGs that you should be aware about are waveform peaks versus underlying ERP components. So remember that from the last slide, that observed waveforms at an electrode are the weighted sum of ERP components. It's all a connection and a certain weight. So naturally, what you're seeing at the electrode is not representative of what the actual ERP components are in the brain. And so the peaks that you see on the electrode waveform are not necessarily the peaks in the components. In fact, it might not be even the same time frame in which they occur. And so it's important to know that when you see an electrode and you're looking at all these peaks and all these squiggly lines, the data on the electrode itself, the wave from the electrode itself, doesn't have any physiological meaning. It's all a combination of what's happening underneath in the underlying ERP components. So waves and peaks and troughs don't really mean anything. There's no physiological meaning. It can be correlated. There's like a chance that it might actually line up and be on the same time scale, but you cannot with 100% accuracy deduce anything significant without doing any sort of further analysis. So here's like a big diagram that I thought it'd be cool to walk through because it really helps you solidify these concepts. So let me explain exactly what we're looking at right here. So if you look at panel A, this is like a waveform that you see off of one electrode. So if you just looked at, if you had 64 electrodes and you took one of those electrodes for like one specific trial, this is what you would see. Now, panel B and panel C are the underlying ERP components. So what you see over here in panel A is a combination of these three components in panel B. So the sum of these with the different weight will lead to this waveform. Alternatively, a sum of these can also lead to the same waveform. You don't know which one it is. These are just hypothetical examples to prove the point that like what you see over here is a combination of different components and different intensities of those components as well. And notice that in this, let's pick B for example. So in B, the first peak that we see here, C1, occurs at 100 milliseconds. However, the peak that we see here in panel A occurs at 50 milliseconds. So if I just saw this on an electrode and assumed that they were the peaks directly translated to peaks in ERP components, I'd be completely wrong. In fact, the component only peaks at 100 milliseconds. So be careful when you look at the data because it really distorts. It's really distorted and it's hard to make significant conclusions. Now, furthermore, what we're looking at in panels D, E, and F are different condition experiments. So you have the baseline, which is a solid black, and you have three different experimental conditions, X, Y, and Z, each represented by dotted lines or dashed lines. And if you look at panel E, for example, you see that the condition actually caused an increase in the first peak and a decrease in the second peak. And if you were, and by the way, in EEG data, we call both things peaks, because because of technical issues, or like it's just been like that, but we just call them peaks. Um, so over here, like I was saying before, you have peaks. You have one peak, an increase in one peak, and a decrease in the next peak right here. And you might be tempted to assume or conclude that there was actually a change in two ERP components because you saw a change in two peaks. But actually, there has only been a change if you, according to the experiment that was done, there was only a change in the C1 component. And it was actually an increase in the C1 component, which caused this change in wave pattern. So just because you see a difference in two peaks doesn't mean they're different in two components. It can be the difference in just one component. Panels G, H, and I are what we call difference waves. So difference waves are, as the name suggests, when you subtract the difference between the experimental condition and the base condition. And difference waves are useful. They tell us a lot about, like, ultimately, like, what we're looking at and the difference in activity between two different conditions. But they only work if the experiment is controlled enough where they only differ in one component. So if you still had the difference in multiple components, the different wave wouldn't be as useful, because the whole point of the difference wave is to isolate components. And so your experiment should be careful and carefully constructed enough and thoughtful enough where you're pretty sure that you're only isolating one component and not any others for this kind of technique to work. So there are different forms of ERP components that are commonly recognized and commonly elicited when we engage in certain activities. So there's no solid naming convention for an ERP component. We use N or P to dictate whether it's positive or negative. So whether the voltage spikes or whether it dips. And it's also followed by a number typically. And the number can represent two things. In one case, it can represent latency. So like latency means the delay between when the stimulus was presented and when we saw this peak. So in this case, N400, which is a ERP component used when we're studying language, we're looking at, it's called 400 because it's present, or it's detectable 400 milliseconds after the stimulus has been presented or after the task has been initiated. The number can also refer to the ordinal position. So P2 refers to the second major peak. It's not relative to time in milliseconds, but it's like the second major peak that we see in this particular waveform. So the P2 component actually in an ERP is the rare versus frequent stimulus. So like there's a famous experiment that's, you know, super basic and super standard. It's called the oddball experiment, where you see a string of letters or numbers or characters that are all the same, and then and then in every random interval you inject that, or you separate that with like something that's unexpected. So you can see like square, 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 circle, square, square, square. And when you see that circle, you're not expecting it. It's something different. That component is elicited. The N170 is another famous component. That's elicited when the stimulus is in our domain of expertise. And what I mean by that is like this goes for everybody. The N170 component is present when we're looking at faces. So as humans, we're trained to recognize faces in an instant, trained to notice the subtleties and subtle differences in people's faces. We're experts at that as social beings. So the N170 is always elicited in that. However, if you have someone who's, you know, familiar with a certain like dog watchers or like bird watchers or dog breeders, for example, that are very familiar with like a certain type of species and their breeds and characteristics, they actually elicit this component as well, even for non-humans. So it kind of suggests the idea that we should have extensive experiences with them and expertise in that domain for it to be elicited in that situation. So there are also, just like how there are different ERP components, there are also different kinds of brainwaves. And you guys might have heard about this already. Our brain is actually constantly doing something, even when we're just sitting still or sleeping. And when we're actually completing a task or when we're doing something, it's actually very hard to look at any sort of pattern because the EEG data is all over the place and there's no determinable pattern. However, it's actually when we're doing nothing, like when we're just sitting there, that we tend to observe these neural oscillations. So starting with beta waves occur at a relatively high frequency, if you see 13 to 30 hertz, and it's when we direct attention towards a particular task. So when we focus our conscious attention towards something is when we typically see beta waves. Alpha waves are present in the initial stages of sleep or even when we close our eyes. So if someone's actually really tired and they close their eyes momentarily, or someone's about to drift off to sleep, we see alpha waves. It's like in that initial period of like zoning out or just being really tired. Theta waves we see when we are in sleep or in some sort of deep meditation. It's often associated with like dreaming and some sort of vivid imagery when we're like transcending the boundaries of our consciousness and our ability to control perception. So like we're aware of it, but at the same time we're not completely in control of it, and that's when we see these theta waves. Delta waves are as you can see like the lowest in frequency, and they're those like big dull repeating waves, and you only see this when you're in deep sleep, and you typically never see it anytime else. So, um, so we've talked about a lot so far. This is another important concept in EEGs that's very relevant when you're collecting data, and and my PI tells me this all the time. But there's no substitute for clean data. So there's a lot that you can do to the data once you've collected it. Like you can average it, you can filter out the noise, you can run all these algorithms on it. But all of those come with certain drawbacks, and that's that they distort the quality of the data. There's like a trade-off that you have to incur. And so the basic like rule of thumb is that there's no substitute for clean data. So when you're trying to collect data or trying to build a brain computer interface or trying to do something, your primary goal should just be to collect accurate data, so you don't have to worry about playing around with it and trying to clean it up after you're done collecting it. So EEGs are recorded on a scale of microvolts and milliseconds. So that makes them very subtle and easy to introduce noise because they're so minute. So what we do is that in when we're collecting data, it's not so much the noise that matters in an absolute amount, but it's all relative to the signal. So how strong is our signal compared to the noise, which is why we introduced the concept of a signal to noise ratio. So a signal to noise ratio is actually the function of the square root of the number of trials. So your idea is to maximize the number of signals or the quality of the signal to the level of noise, right? You want to have a high signal to noise ratio. But because it's a function of the square root of the number of trials, you have to actually quadruple the number of trials to double the ratio because the square root of 4 is 2. So if you have like 20 subjects, quadrupling them, like getting to 80, is actually a lot of work. And so you can easily see why increasing the number of trials to increase the ratio is not always feasible. And so your best bet is to try to get the cleanest data that you can in the first place. After you have collected the data, one big way to reduce the noise is by averaging it. And here's an intuition for why averaging it would work out. So let's say you have this big ERP component, right, that goes like in a big wave. And anytime you collect data, you'll encounter some noise. So like when you're in this big wave, there'll be like little ups and downs, and every person will differ. But if you have like maybe like a +2 of microvolts and another person has a -2 and someone has a +3 and someone has a -1, all of those little deflections that you see will eventually average out to 0, but the overall pattern will still remain. So if you look at this data right here, here are individual trials from 8 different people. So on the left is like person 1, person 2, all the way to person 8. And then average data is like just person one and two, all the way until you encounter the 8 people. So if you see the graph, the average graph tends to get a lot smoother and compared to the individual trials. You see a lot more jagged bumps in the individual trials versus in the average one, it's a lot smoother, and the pattern becomes a lot more discernible until you get to the last one actually, and you see like that sustained level of activity from almost 300 milliseconds um, to about 600 milliseconds, compared to the individual ones where you saw like a lot of activity and it was hard to even find a pattern. To someone who like isn't familiar with this data, it might even look like in the last waveform where you see these circles, it might even look like I mean, like, what is that? Like, that doesn't look like a distinctive pattern or a waveform at all. And that's part of just having experience with it. But if you use machine learning models to train it, they're better at detecting patterns and detecting the intensity of and the differences between the stimulus presentation and baseline recording. And so that would help in detecting patterns like that. So we covered a lot of new material today, and a lot of it's pretty dense in terms of complexity, and we basically only have scratched the surface of it. Like this stuff is, you can go way deeper into it, and there are a lot more nuances that come up when you're actually recording and collecting this data. So I just want to do a quick overview of what we've done, and hopefully that should help you guys, and we can end it for today. So we first talked about current versus voltage, right? Remember, current is the amount of charged particles moving through a conductor, whereas voltage is the potential of electric current to flow from one location to another. So voltage necessitates two things. It's always relative between two different electrical points. We talked about EEG systems. We talked about the electrodes, the gel substances, um, um, how it's different in a BCI, in like the one from Zito that we saw versus the traditional clinical EEG system. We talked about active reference ground, how ground is always necessary when you measure an electrode and its electric potential. It's always between the electrode itself and the ground. But if you want to measure the difference in an active electrode versus a reference electrode, it's A minus G and R minus G, which then gives you A minus R. We talked about the superposition problem, which is super important, which is the fact that all the electrical activity that we see from an electrode is a combination of ERP components and their respective weights to various degrees of intensity. We talked about waveforms and ERP components, and how a waveform, a peak on the electrode that we pick up is not exactly a peak in the components, nor does it really tell us anything about the components at all. We talked about different types of brain waves, right? Alpha waves, beta waves, theta waves, delta waves. And last but not least, we just talked about signal noise ratio and averaging, and how it's important to maximize the ratio instead of just an absolute measurement of the noise, and how averaging can help reduce that. But then again, there's no substitute for clean data at all. So clean data is first priority. So that's it for this module. Hopefully you guys were able to grasp. This was fairly more complicated than the first two, I would think, at least in my opinion. That's what it seemed like. So if you guys have any questions, feel free to email any one of us or just hit us up on Facebook. Also, please be sure to complete the self-evaluation, and we'll be out with the next module next week.